Good day everyone. Today I want to present our paper which has a title understanding the mechanism adsorption of heavy metal by free and immobilized Chlorella protein. This paper was written by Savira, Aulia, Putri, Lidia, Pradista, Putri, Handoko, and me. We are from Universitas Airlangga, Indonesia. For the first part is introduction. So, nowadays, with a high population to affect the high water from the human activity, several environmental problems have been caused by toxic contaminants like heavy metal from human activities, polluting the environment and the surface water resource. Heavy metals pose a serious threat to human health because of their chemical characteristics, such as the toxic effect and tendency to accumulate in the food chain. If they are found to be above the tolerance level, they can cause the accumulating poisoning, cancer, and the brain damage. Microalgae that has ability to remove the heavy metal, for example, the species of Chlorella. Application microalgae in remediate polluted water has several methods. Microalgae can be applied in suspension or free put in water, and the other method is in treatment in polymer, or well known as the immobilized method. Suspension method is applied with a freely microalgae in water with a high concentration of heavy metal, and a method of immobilization that is most commonly employed in treatment and encapsulation in polymers. The mechanism of removal of heavy metal by two kinds of method is not well explained in several papers. So, of this study is to explain about the different mechanism of removal heavy metal by free and immobilized Chlorella vulgaris. First, we have two types of methods. For the free methods, free suspension methods, we can put a certain volume of the free cells of Chlorella was put in water with a high concentration heavy metal. And before this, we need to do the culturing process. And the other part is immobilized method. So this part is prepared with the entrapment the chlorophyllis with polymers such as alginate, and then conducted to water with a high concentration of heavy metals. Both of them, the microalgae was conducted at certain times. So for example, 60 minutes, 120 minutes, or etc. So it depends on the purpose of the experiment. So this is like the results of our experiment and compare with the other research results. So we using cuprum, and the other like Chen et al. In 2016 using the chrome, and Elsek using the cadmium, and Ahmad et al. Using the iron, mangan, and zinc, and Kwartia Kozlovska using the another metal, the lead. And we have similar microalgae using the Chlorella vulgaris. We also have different methods like the free suspension or immobilized. For the application, put the Chlorella vulgaris with heavy metal. So from all the table, all the our research and the other researcher shows that for the free suspension, usually the removal of metals is around 70%. However, the immobilized have the more higher, is around the 80% for the removal of different kind of metals. So it shows that the removal of heavy metal with the immobilized Chlorella vulgaris or the other species has a higher efficiency than the free cell Chlorella. The existence of polymer as entrapment material for chlorella support the capability to remove the pollutant. So the mechanism of adsorption by the free cells, the Chlorella vulgaris, we know that chloroplast has is the part of the Chlorella vulgaris has uh, the function that can remove chloroblasts was produced by Chlorella vulgaris boost to the ability to absorb the heavy metal and also to reduce the metal. So this is the illustration how that the chloroplast exists inside or produced by the Chlorella vulgaris. The other part of the chlorella also has another function is the cell wall. So the cell wall is had have a part for the bioabsorption of heavy metal because due to the ion exchange method where the ion present on the algal surface like magnesium and then sodium and they are displaced by the metal ions and the complexation between the functional groups and metal ions also have a part how the metals can remove at the surface of the cell walls. This is the illustration of vulgaris. So the green line show the cell wall is surrounding about and the surface of Chlorella vulgaris. So another part is the environment conditions. So the environment removal of heavy metal in aqueous solution was done under the optimum condition for the pH, temperature, time, agitation speed, pH, solvent dose, and the metal concentration. So this is for the example. So by increasing the pH value, the additional the ligand of the chlorella, such as the amino or carboxyl groups, become exposed, and which to produce the negative charge. So that's why it could be occurs the ion exchange. Uh, this is next part is about the mechanism removal by immobilization, vulgaris. So in this part we will talk about also similar about ion exchange. So we know that the main advantages of immobilization also include the prevention of the biomass washout from the process and enhancement operational flexibility. So with the entrapment of the microalgal cells or the chlorella cells in the small confined space, this is also can trigger the interaction for the between the chlorella and the pollutant and enhancement of nutrient uptake. So the next process would occur is the major cation exchange. During the bioabsorption process was the sodium ion from the sodium alginate cell gel. So this is the material for the polymer that used for trap the chlorella along with the calcium ions. So the peak signal of this cation may be reduced or eliminated if they are exchanged. So immobilized cells also have a higher metal absorbing proficiency compared to the free cells. So this part, the size of bits, so the bits is the name when the chlorella already entrapment by the polymer material, and the size of bits is used for the biomass immobilization, and this is important factor during the bioabsorption process for the immobilization methods. As the conclusion that we can see that the chloroplast and the cell wall of Chlorella vulgaris has the main function to absorb heavy metal by free suspension of Chlorella vulgaris. And environmental conditions also affect the bioabsorption process. And the cation exchange between metal ion and polymer material for the Chlorella vulgaris in immobilized system also has important factor. And Also, the size of beads also affects the absorption process in the immobilized system. Thank you. That's all of our presentation, and thank you for your attention. And if you have any question, please feel free to contact me in this email, nurio@fstunair.edu. Thank you very much.